All right, great. My name is Steven. I'm a master's student here at the University of Wisconsin-Madison, biomedical data science. And my talk today is on integrating Perturb-seq and GWAS summary statistics to identify genes for human complex disease. So I'll start with some motivation and then get into our model a bit, and then show our model in real-world data applied to inflammatory bowel disease and coronary artery disease, and end with some summary. So the motivation behind this is some of the limitations that currently exist in GWAS and TWAS methods. Jiacheng just described some what GWAS is. It finds associations between SNPs and phenotypes, but linking these signals to mechanisms of disease and complex disease is historically very difficult because we find many SNPs across the genome with very low effect sizes. So TWAS identifies associations between genes and phenotypes instead of SNPs and phenotypes. And it does this through the regulation of gene expression. So it uses a reference panel of genetic data and gene expression data from donors and healthy tissues, and then trains imputation weights. And then later you can use genetic data from other individuals to predict their gene expression for genes and then associate them with traits. But importantly, a limitation of both of these methods is they cannot find associations for constrained genes because their population level, they use population level cohorts. So if there's no variation in the population of gene expression or of SNPs, then they're not going to find any association. So what is Perturb-seq and how can it potentially help us? In Perturb-seq, we perturb a set of genes using CRISPR-based gene editing and measure the effects on downstream gene expression levels using RNA sequencing. Our hypothesis is that we believe that there are a small number of constrained genes that regulate many of the weak Perturb-seq or many of the weak GWAS signals. And so we can potentially use Perturb-seq to find these constrained genes by integrating that data with GWAS data. So Perturb genes can be under selective constraint, so we can find these genes that GWAS and TWAS cannot find. But in Perturb-seq, we have RNA sequencing data and gene expression, and we need to somehow associate these effects with the phenotype. So that's what our model attempts to do. Our model's attempts to estimate the effect of the focal gene X on the phenotype Y through these intermediary mediator genes, G. So gene X here is a gene that is perturbed, and from Perturb-seq data, we get effect estimates on how perturbing this gene affects the gene expression of many downstream mediator genes, G. And then from TWAS, we get an effect estimate on how the gene expression of gene G affects phenotype Y. So our model uses attempts to look for synergistic regulation through these mediator genes G. And we filter these mediating genes by ABC score and TWAS p-value to attempt to get genes, because from Perturb-seq we get many tens of thousands of downstream genes that are measured for each perturbed gene. So this filtering attempts to keep mediator genes in the middle that are important in the cell type that we're looking at for the phenotype that we care about. So we applied our model to a couple of real data sets. This Nature paper found that there are many GWAS SNPs across inflammatory diseases, are most significant in IBD. And through looking at ETS, EPTL data in macrophage data types, they're able to associate this GWAS peak with the gene ETS2. And then they later did additional functional follow-up experiments to show that they increased gene expression of ETS2 in macrophage cells and induced an inflammatory state through this low-dose LPS, and then later collected single-cell RNA sequencing data downstream. So this summary statistics from single-cell RNA sequencing data looks similar to what we would get from a Perturb-seq data set, but it's just looking at this one ETS2 gene that they studied. And And they had 3 different states of LPS dosages, so 3 different inflammatory states of macrophages that they collected data on. So we used this data, and then we also had to run TWAS to get the other set of data for our model. We used EQTL summary statistics from inflamed macrophages from this paper, and trained TWAS weights using otters, which allows you to train TWAS weights without individual level data, but just with EQTL summary statistics using various PRS methods, and then run a normal TWAS with those imputed weights. And then normally they get a bunch of p-values from the TWAS and then do an ACAT test to get a final p-value. Here, since we need the effect size from TWAS, we ran our method using each of these TWAS methods alone. And then after running our method, we used an ACAT test to get a p-value. We got a p-value of 0.01. And the effect size is positive for all dosage and TWAS methods that we tried for ETS2. And so a positive effect size from our method means that ETS2 overexpression leads to an increased risk for IBD, which is what they found in their paper. So we use this as a positive control to show that our model works to find this gene. And then we tried to find some novel results in coronary artery disease. There's this large Perturb-seq in endothelial cells that attempted to look at coronary artery disease. So they did CRISPR-I on genes near the 306 CAD GWAS loci in endothelial cells. They did this because 75% of GWAS signals for CAD are not associated with circulating lipids like cholesterol, which can be more easily explained. And so they attempted to explain many of these GWAS signals through using endothelial cells because they're important arterial cells which have an impact on cholesterol, smooth muscle response, and blood clotting, and are potentially interesting for coronary artery disease. So this is their study. And then for a TWAS to get gene expression and CAD association, we used the largest publicly available coronary artery disease GWAS, and also used all of us GWAS that we ran ourselves. Both of them are largely of European ancestry. And then we used the TWAS fusion method using 10 relevant GTEx V8 tissues with sample sizes ranging from about 200 to 500 donors per tissue. So we find 4 FDR significant genes in coronary artery disease from our model. Here they are, and on the right is the QQ plot that shows the p-values resulting from our model. And then we attempted to replicate these in independent GWAS cohort from all of us, and we found that the effect sizes all had the same effect direction and less power, and the p-values were all statistically significant. So we were able to replicate all of our results in all of us. Here's an example of the gene with the lowest p-value, and shows the correlation between Perturb-seq and TWAS signals after we filtered for the downstream genes using ABC and TWAS p-value cutoff. So this positive effect direction means that perturbing MRPL33 and reducing its gene expression leads to an increased risk for coronary artery disease. And if we just take MRAS as an example, a positive TWAS score means that an increase in its gene expression will lead to a higher risk for coronary artery disease. And a positive Perturb-seq effect means that decreasing MRPL33 leads to an increase in gene expression of MRAS. So this large synergistic effect that we find for many genes leads us to think that perturbing MRPL33 leads to increased risk for CAD. So in summary, we've introduced a new statistical approach to integrate GWAS and Perturb-seq data. We're attempting to discover genes that regulate many of the weak GWAS signals that we see and explain some of these GWAS signals. And our model was able to successfully identify the ETS2 signal for inflammatory bowel disease in macrophages. and we find 4 FTR significant results for coronary artery disease, and replicated all of these results in an independent GWAS from all of us. And so this is an ongoing project. We have some next steps, which are to dive deeper into some of the genes that we found that were novel for coronary artery disease and attempt to biologically explain them. And then we also plan to apply our method to additional Perturb-seq data sets and diseases. We've recently gotten some new Perturb-seq data sets that we're excited to apply our model to. I'd like to thank all of my collaborators, especially Professor Q and many of the lab members, and I appreciate your time today. Thank you. Sure.
So how much does the does it have to be Perturb-seq, or can it be other bigger perturbations, some variation data that you're actually using? And how much does the Perturb-seq data have to be relevant to the system that you're looking at? Like for example, you looked at the CAD data set, but not all data sets can have an associated Perturb-seq data set. So
right.
Can you use like a whole collection of Perturb-seq data sets and leverage that?
Yeah, we plan to use like larger Perturb-seq data sets and maybe run like a phenome-wide scan for multiple different phenotypes that we run through TWAS. I think generally the cell type needs to be somewhat explanatory for the disease, and the cell type between the Perturb-seq data set and the TWAS tissue that's used needs to be ideally a perfect match, because then the biological explanation just makes a lot more sense. But yeah.
Interesting. So, other questions? What are you assuming about the Perturb-seq data set? Like, because there is a lot of noise in Perturb-seq data sets, and are you assuming that, this is your timer, are you assuming that the gene that is to be perturbed is somebody's telling you because that was the experiment that was done, or are you doing something more?
Yeah, so
cleaning up
the results in like summary statistics that are provided at the end of Perturb-seq are like single cell RNA sequencing, where it's differentially expressed of the gene was perturbed and no gene being perturbed, and there is a lot of noise and like issues in the data for sure. Our model does assume that the effect estimates are unbiased. But yeah, that's definitely something we could look into more in the future and be able to work with noisier estimates.